All right, welcome back to EBE. Still on this human evolution question. What we have here are lots of nucleotide sequences from gorilla, human, and chimp. And what we're going to do is look at these sequences and determine how similar they are. More similarity in common sequences, the more related or the more recent a common ancestor these two taxonomic groups have. So if we look at different parts of the genome, and I've got a few listed here in this table, it's probably worth going through these so that you can remember them. If this is our model for a gene, this is composed of a regulatory region and a coding region. In the case of most genes, this is protein coding, but keep in mind that some genes are RNA coding. The transfer RNA genes, the ribosomal RNA genes, etc. So within the gene itself, we could define particular regions. So there are regions that code for, say, protein, and regions that don't. These would be the exons are the coding region, are the coding sequences, and introns are sequences between the coding regions. These are being edited out during post-transcriptional processing. So within this table here, intronic would be the I's, the coding sequences are the E's. Then keep in mind within the coding sequences, we could have differences in nucleotide sequence that occur at the third letter. So, for example, if this is the RNA, right, that would code for methionine quite obviously. And if you remember from MCB, a lot of these codons, the third position is silent, which means CCG could code for the same thing as these sequences. Makes sense? In which case, changes in the third letter don't change the amino acid. So these would be what we referred to as synonymous substitutions in MCB. Some changes in nucleotide sequence in the gene result in unique codons in the RNA, which result in unique amino acids. These are referred to as non-synonymous substitutions. Okay? So we're looking at these sequences based on what they do. Let's look at the introns differently than we look at exonic sequences, the ones that actually do the coding. Then we could also look at non-coding sequences that aren't in genes but are between genes along the chromosome. So that covers this one, and this one. The X chromosome and the Y chromosome they have here, because the X and Y chromosome do not cross over and recombine. During prophase 1 of meiosis, the autosomes do crossing over and recombination, but the sex chromosomes don't. So the idea is any differences in sequence would be due to mutations, not due to crossing over and recombination. Then, the last one to talk about here is this pseudo gene. So a pseudogene is a gene that is a copy of a gene within the chromosome. So I could have in the DNA molecule itself, let's say 3 copies of the same gene. These copies arise through meiosis. You'll talk about that more when you take genetics or human genetics, whichever one you take. But these molecules here, these short sequences are copies. So this is the same gene that's been copied over within the DNA molecule. Now, because the organism needs protein from this gene, if something were to happen to this gene, it wouldn't affect protein production because I still have 2 copies. So if we look at this gene, we know that this regulatory region helps to get this gene transcribed, right? The transcription factors bind to particular sequences within the regulatory region. I could have a mutation here, though, that destroys these promoter sequences, and that would mean that this gene is no longer transcribed. The information is still there to build a particular protein, but the cell has no ability to access that information. That's what a pseudogene is. Sometimes pseudogenes are called fossil genes. The idea is they're genes left over from back further on in the lineage. A mutation disabled the gene, right, made it so that it couldn't be transcribed, and now you have this kind of fossil gene hanging out in the genome. We call those pseudogenes. Okay? Now, the way that the ancestor of all 3 of these, right, the idea is that I have an ancestor to all 3 of these, and then human, chimp, and gorilla. That was the consensus phylogenetic tree that we built last time. What this suggests is that the human and the chimp should have more similar DNA than the chimp and the gorilla, or the human and the gorilla. And the idea is that the DNA molecule here has been copied more times to get to the present than this DNA molecule has been copied to get through to the present. Now, any differences that I see in sequence among these 3 groups is due to mutations that occurred along these different lineages. The DNA is picking up mutations every time it's being copied. So the idea is any differences we see are due to these mutations. The more differences in sequence that we have, the more distantly related they are because I've accumulated more mutations because I've had more time to copy the molecules. Okay? So if we look at this chart, let's just start out with something that we know should be relatively unaffected by mutations, and that would be non-coding intergenic sequences. Any mutations that occur out here in this non-coding intergenic region shouldn't have any effect on the organism, and therefore these mutations are what we say is neutral with respect to selection. These mutations have no effect on the individual, and therefore whatever mutations occur accumulate within the molecule. So this says that the genetic distance between chimps and humans is 1.24, right? Which would be 1.24%, because this is percentage sequence divergence. So that means roughly 98.76 out of 100, or 9,876 nucleotides out of 10,000, are similar between a chimp and a human if we look at these non-coding intergenic regions. Okay? If we look at pseudogenes, we also know that pseudogenes are not under selection. That's about 1.64% difference. And if we look at synonymous substitutions, that's about 1.11% difference. But the non-synonymous substitutions is only 0.8, and the intronic is 0.93. And the most obvious explanation for this is that if a mutation occurs that doesn't change the amino acid that gets placed, then selection really doesn't care. On the other hand, if you change an amino acid at a certain position, you're giving natural selection something to work on. And some of these mutations that occur reduce the fitness of the individual, which decrease the likelihood that that sequence gets passed on. So it's almost like what we're looking at in these 2 instances are mutations plus selection. So because selection is working on these, it's underestimating the difference between chimps and humans, because it's saying that they're only 0.8% difference, whereas the pseudo gene is roughly giving us twice as much difference. But in any event, if we look at a particular estimator here, the chimp and the human, the difference is 1.64%. And the difference between a gorilla and a human is 1.87%, or 1.87 nucleotides out of 100. And then the difference between the gorilla and the chimp is 2.14%. Okay, so there's a lot of sequence similarity between humans and chimps, and even between humans and gorillas. And I say this because from an outside perspective, if we asked, hey, here are two species that are very closely related, the eastern bluebird and the western bluebird, or these two species that are very closely related, our own blue jay plus the Steller's jay, we could ask, well, how different are these? Kind of like the game we played with HIV and asked, well, how different are they? Well, the difference between the eastern bluebird and the western bluebird is 10%, right? There's 10% difference, which means that 10 out of every 100 nucleotides are different when you compare these two birds, and 10 out of every 100 nucleotides are different when you compare those two species, which suggests that the difference genetically between human and chimp is much, much smaller, at least genetically. And that bothers biologists because we like to think that we're a lot smarter than a chimp. So the next step was to ask the question, well, if the sequences are all that similar, is there anything else that could possibly explain the difference among chimps and humans, at least phenotypically? This experiment was an attempt to answer that. And the argument is, yes, we're very similar genetically, but the small amount of genetic differences result in large changes in expression differences. So the idea here is the chimp and the human have the same gene. Genetically, 1.6 nucleotides out of 100 are different, but they're very closely related sequences. But if one of these differences lies here, perhaps what this does is increase the rate of transcription relative to what it is in the other species. In other words, the differences between humans and chimps, albeit are small genetically, they have large effects on expression. How quickly the gene is expressed, how much protein accumulates after this expression. So the idea is they looked at the blood, the liver, and the brain, and they're looking at differences in the expression of certain genes. The length of the bar indicates differences, and the idea is that particular genes that produce proteins that are active in blood cells or in liver cells, that's about that same number that we've dealt with before, right? Whereas in the brain, there are these huge differences in expression. And that's where we are currently, is arguing that humans, we have the same genes as chimps, but we express them in different ways, which allow for us to have this huge difference in intellect. Okay? All right, so that gets us to the first piece of this. Humans are related to the African great apes. We have a common ancestor with the African great apes. We're more closely related to chimps, at least in terms of sequence similarities. Okay? All right, then we can say, well, once this lineage, right, here's the gorilla, chimp, and human. So in here we can ask, well, once this early hominid appeared on the planet, what does this lineage look like? Kind of the hominid lineage, not the African great ape lineage, but the lineage that arose after the first hominid showed up. So that's what we're going to talk a little bit about here. So we'll do the same thing we did before. We start out with these synapomorphies. We look at similarities in traits between putative organisms or putative species, and then we ask about similarities. So for the early hominids, it's not like we have any individuals that we can look at, like the extant gorillas or chimps. We can look at them and compare. But we don't have any other hominids around besides us. There's just Homo sapiens. The other members of the genus are all fossils. So we have to then look at things that fossilize well, which would be bone. So we're looking at skulls and teeth. So here are a few of these early ones. There's the Australopithecus genus, Kenyanthropus genus, Ardipithecus genus. There's some Paranthropus here. And then there's some Homo ergaster, Homo habilis, Homo rudolfensis, Homo sapiens at the top, Homo neanderthalensis, Homo heidelbergensis, and Homo erectus. Now, what you could see, at least trait-wise, is differences in the elongation of the skull and how much this frontal part of the face kicks out this way or is pushed more upward. So if you look at like the Kenyanthropus, that angle on the skull is very large, whereas a Homo sapiens skull, the front of the face is more pitched like this. Notice also that we don't have complete skulls for a lot of these. So a lot of this is kind of inferential based on what you have. More pieces are found every day. But the idea is that we're mainly working off skulls, and we're working off of partial skulls. Okay? So you see that kind of here in this Paranthropus. Look how pitched out that is. It looks almost like a monkey. Okay? And then by the time we get to the Homo genus, the face is pushed more up like this. Okay? Homo sapiens at the top. Look at that. That's a 30,000-year-old skull. Here's Homo neanderthalensis, heidelbergensis, and erectus. All very closely related to us, at least in terms of trait similarities. Okay?